All right, so today's topic is looking at mitosis and cytokinesis. Remember, mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm. Mitosis occurs first, and there are 4 phases, and I have underlined the first letter so you can remember the acronym PMAT for prophase, metaphase, anaphase, and telophase. Those are the 4 phases or the 4 stages of mitosis. Now, most of the changes occur during prophase. Metaphase, anaphase, and telophase are pretty straightforward, pretty easy to remember, but prophase, there's a lot going on.
In prophase, chromatin containing the DNA is condensed into structures called chromosomes, which are joined in identical pairs at the centromere. Spindle fibers appear from the centrosomes, and the nuclear envelope and nucleoli disappear.
Okay, so it doesn't sound like a lot, but there's a lot going on. So remember that the second phase of interphase called the S phase, I know it's lots of phase. It's like phase for any drag superstar fans out there. Anyway, during the S phase, DNA is copied. So during prophase, all of that DNA, remember, hold on. No, go back. Oh, sorry, I just got to take attendance here for a sec. Okay. So, and that window, let's go down. All right. Okay, so during interphase, you have the G1S and G2 phase. The S phase is when DNA is copied. Now, if you remember the analogy that if the nucleus is the size of a basketball, one full DNA molecule is 64 kilometers long. So during the S phase, you're going to have twice as much. The first thing that happens when prophase starts is that the DNA starts to coil and condense into chromosomes. So that's number 1. Okay, waiting for my iPad to connect. There we go. All right, so number 1, the DNA coils into chromosomes. Number 2, the nucleus starts to disappear. You can see the nuclear envelope here, and now it's represented by a dotted line because you're going to need the entire space of the cell to organize the chromosomes and have them separate. Now, you have 2 dark spots, which you can see here. Got that dark spot and this dark spot. These are called centrosomes. They start to appear. Now, in animal cells contain centrioles, and centrioles were part of chapter 4 when we looked at that. Centrioles are cylinders that are at 90 degrees to one another. In my fantastic art skills, you can see there. That was sarcasm. For anybody who doesn't know what sarcasm is, it's when you don't really mean what you're saying. So these cylindrical bodies. Oh God, I should just give up. Okay, anyway, there are 2 cylinders that are at 90 degrees to each other. Okay, they only exist for animal cells. You do not find centrioles in plant cells, but centrioles in animal cells are part of the centrosomes. Okay, there's again a lot of vocab I'm like throwing at you, which is why I wanted to separate yesterday and today. All right, so we have condensed chromosomes. Obviously, those come with the centromere. The nuclear envelope starts to disappear. Centrosomes appear and start moving towards opposite ends of the cell. Okay, as they move to the opposite ends of the cell, you have these fibers which are going to form. Some of these fibers are going to connect from one centrosome all the way over to the other. Some of them are going to be connected only to one centriole and reach about halfway. Together, all of this whole thing, so the centrosomes plus all of these fibers are called the mitotic spindle. Okay, so as the centrosomes move to opposite ends of the cell, you have these spindle fibers that are made of microtubules. If you remember microtubules from chapter 4, if you don't, you can go back and review. microtubules are part of the cytoskeleton that are made up of a protein tubulin, and they're called microtubules because they are hollow on the inside. So these long fibers that are hollow on the inside. There are two kinds of fibers, as I mentioned. You have kinetochore fibers and polar fibers. And now it's a little bit easier to remember the difference between the two. If you know what polar means two opposite ends, right? So the polar fibers are the ones that go from one centriole all the way across to the other centriole. So you've got some fibers that go all the way from end to end. And kinetochore fibers. Now, again, remembering what the prefix "kine" means, that means something to do with movement. So the kinetochore fibers are going to be involved in moving something. Well, what are we doing? What is this the process of? This is the process of mitosis, dividing the nucleus. What does dividing the nucleus entail? It entails organizing the DNA into chromosomes and making sure that each new cell has exactly one set of chromosomes. How are you going to do that? You're going to have to move the chromosomes. You're going to basically line them up all in the middle, and then you're going to separate them. That's going to require some kind of fiber that's going to attach to them and move them to opposite ends of the cell. So the kinetochore fibers are the ones that go only halfway. They come out halfway. They're going to attach to the centromere of the chromosome, and they are going to separate the chromatids. Okay. So on the centromere, which you see here, you have this structure called the kinetochore, and that's why they're called kinetochore fibers, because the kinetochore fiber is going to reach here and separate them. There we go. Going to separate the chromatids, right? So the spindle fibers, specifically kinetochore. Oh, that's hard. It's a hard color to read. Let's go with the dark blue. Yeah. Kinetochore fibers are going to attach to the kinetochore at the centromere. Okay, so that's prophase. So let me go back one. Just to summarize what's going on in prophase, DNA condenses to chromosomes. 2, nuclear envelope disappears. If I could learn how to spell, that would be great. Envelope disappears. 3, centrosomes appear and move to opposite ends. And 4, the spindle fibers form. Okay, so all of that happens. Oh, thanks, Bluetooth. That's awesome sauce. All right, never mind. Well, I got that on video anyway, so you can always go back and watch it. So those are the main events that happen during prophase. As I said, the majority of the changes that occur during mitosis happen during prophase. So metaphase, anaphase, and telophase, if we follow PMAT, prophase, metaphase, anaphase, telophase, most of the stuff, the quote unquote stuff, happens during prophase. So metaphase, anaphase, and telophase will be much easier as far as how much change is going on.
In prometaphase, some of the spindle fibers attach to the chromosomes, causing them to move. In metaphase, the chromosomes that are now attached to the spindle fibers line up along the midline of a cell.
Okay, so I don't separate out prometaphase. I just make it part of prophase. Metaphase. Meta means middle. Not like the new name of what do you call it? Facebook. Bleh. Trying to, I don't know, make themselves look better or something. They have this thing called the metaverse now, and I keep thinking like Zuckerberg, bro, like everyone has been doing virtual meetings and virtual classes for like 2 years. Like nobody wants to be in virtual reality anymore. Like I crave interaction with real people. But okay, you go on and make your avatars. So like whatever. But meta in science means middle. So that should be easy to remember what happens during metaphase. Literally the only thing that occurs during metaphase is the chromosomes line up in the middle. Now, why do chromosomes line up in the middle? Because then it's easy for the kinetochores to attach to the centromere and then separate all of the chromatids from each other. So here again, we have the kinetochore that's part of the centromere. The kinetochore fibers are going to attach, let's say, even before onto like different chromosomes. We got like a purple one, whatever. So you have these kinetochore fibers that are going to attach. Actually, they should be coming from the same place, sorry. Centrosome, kinetochore fiber is going to attach to the chromosomes, and actually the kinetochore fibers are going to move them to the center, okay? Once they're in the center, they're going to hold them there. Now, once that occurs, this structure where the chromosomes all line up is called the metaphase plate. That is something that I know is not, for some reason, not mentioned in this textbook, but I have seen it in other textbook and on exams. So it's just called the metaphase plate. And here you see the centrioles, these cylinder structures, and you've got your chromosomes lined up in the middle. Now, lining them up in the middle, I was saying before, the reason why they line up in the middle is to make it super easy to separate the chromatids so that you know that exactly 1/2 is going on one side and 1/2 is going on the other side. Now on to anaphase.
Anaphase, the spindle fibers pull sister chromatids to opposite poles of the cell.
Okay, that's it. Anaphase, you're just separating them. So the chromatids are separated at the centromere, and they're going to move centromere first to opposite ends. And finally, we have telophase. After the chromosomes reach the opposite ends of the cell, the spindle fibers are going to disassemble. The chromosomes will start to uncoil, and new nuclear envelopes are going to start to form around each new set of chromosomes. Okay, so you kind of, in telophase, you're kind of reversing what happened during prophase. So you don't really have to remember both pro and telophase. If you just remember one of them and you just know they're opposite of each other. So in prophase, chromosomes condense, DNA condenses into chromosomes. So in telophase, the chromosomes start to unravel and relax a little bit to form chromatin again. What else did I say? The nuclear envelope starts to disappear in prophase. So in telophase, they start to reappear. But now you have to remember that there are two nuclear envelopes. I probably should use a color other than white, because you have two new nuclei because you're forming two new cells. Number 3, the centrosomes appear. So for telophase, they start to disassemble or disappear. And finally, 4, you have the mitotic spindle, all those polar fibers, the kinetochore fibers that start to form. In telophase, they're going to, again, they'll start to disassemble because you don't need them anymore. Oh, and my computer is having issues. Oh dear, what is that supposed to be? Okay, you have to give me a minute. My computer's frozen. Nope. Okay, there we go. Really? It's a video? Oh yeah. See if I can do this again. Nope. Oh, here we go. Okay, so we're inside the cytoplasm. What you're looking at now are the mitochondria. It's mitochondria. Here's Golgi. You can see all of the stuff going to the Golgi to get exported. Here's a nuclear envelope, nuclear pore going inside. And you see the chromosomes start to form. There we go. Nuclear envelope disappears. There's the centrioles, those cylinders that I talked about. So you see that there's no nuclear envelope at all. Metaphase, they all line up in the middle. Here's your mitotic spindle forming. Kinetochore fibers attaching to centromere, metaphase, and ready? Oh, anaphase, separating all of the chromatids from each other. And new nuclear envelopes forming. Still understanding that this is still one cell. We have not done cytokinesis yet. Here comes cytokinesis. In an animal cell, the cell membrane starts to pinch off in the middle until finally you have two new cells. It's going to be a little bit different for plant cells because plant cells have a cell wall which is inflexible. So you can't pinch in on the cell wall, but you can for animal cells. Here's a really good representation of everything except for telophase in one slide. So prophase, how do we know it's prophase? Because you can see the chromosomes take up almost the entire cell. So there's no nuclear envelope. The cell is this size. Okay, these are plant cells, by the way. How do I know they're plant cells? Because they're square. Okay, plant cells are often not round. Metaphase, obviously you see all of the chromosomes lined up in the middle. Interphase is just a regular cell that's not undergoing any division whatsoever. So this is also interphase, this is interphase. Everything that looks kind of normal. Where you don't see chromosomes, okay? The big clue that you're going into mitosis is that you see chromosomes. And here's a really characteristic picture of anaphase where you see those kinds of like V shapes that are sticking out. Those are the chromatids that are being pulled to opposite ends of the cell. Now we have cytokinesis. Again, cyto meaning cell, kine meaning movement, and cis meaning process. So it's a process of moving the cell, or the cell is moving. This is the division of the cytoplasm. And like I said, it's going to occur differently whether you're a plant cell or an animal cell. I would say exclusively or only because of the cell wall. So in animal cells, what happens is a cleavage furrow forms. I don't know the actual definition of cleavage, but I know that both breasts and your butt has cleavage, because obviously you look at this picture, which is an actual cell going through cytokinesis, and that's what we call cleavage definition. But I don't know how you would describe a sharp division or a split. Okay, so that explains why your butt crack is also cleavage, because it's where it splits. And furrow, the word furrow, I think implies like going down or something, because they say when you furrow your brow, your eyebrow, that's when you make like a mean face. Furrow. It's really interesting to me. This is why I could never be an English teacher, because I can't explain anything. Like I know how to speak English, but like if somebody asks me, well, why do you say it this way and not that way? I'm like, I don't know, just because that's how we do it. And that's like explaining a lot of words. Like I know what they are, but I don't know how to explain what it is. So yeah, a furrow, or in this case is a noun, a cleavage furrow, a long, narrow trench. And a trench, right? When you dig like a long ditch, something like that. So that makes sense because it looks like a long ditch. Okay, yeah. Yeah, I would really suck as an English teacher. So don't ever ask me questions about like grammar rules or anything. I am completely capable of like proofreading and telling you how to say things and how to write things. But when you ask me why, I'm like, I don't know. Right, so then plant cells can't form a cleavage furrow because the cell wall is inflexible. So instead, what you have is a cell plate starts to form, which you can see right here. You've got your newly formed nuclei, and now you have basically a bunch of vesicles that go to the center and start basically building a wall. I think Donald Trump would be proud. Build that wall. Just in this case, you're using it to separate two new plant cells. So to go in a bit more detail, so for cytokinesis in animal cells, here you see a nice cross section. So if you look at two cells and you slice it to look at them from the side, what you're looking at here is from the top down, right? So the cell membrane, because it's flexible, it can start to pinch inward. That's creating the cleavage between the two dividing cells. Eventually, because it's occurring from both sides, like you can see here, top and bottom, eventually they're going to meet in the middle. And this is all due to the action of microfilaments, also part of the cytoskeleton. Okay, microfilaments. Again, in plant cells, the Golgi apparatus is going to send a bunch of vesicles to the center of the plant cell, and they're going to fuse together, kind of like bricks. If you think about how you build a wall, you build it with bricks. And so if you keep combining all of these vesicles together, eventually it's going to separate one side to the other side, and it will now separate the two cells. All right, so that is mitosis and cytokinesis. The last thing I want to talk about is how does your body control when this happens and how often it happens and which cells can divide and when does it know to stop dividing. There are measures of control, and when your body loses control over cell division, that is when you get cancer. So cells spend most of their time in interphase. The question is, what triggers the cells to start dividing? In eukaryotes, the regulation comes from proteins. There are specific proteins that are made whose only job it is to check to make sure that the cells, 1, are healthy enough, 2, they're big enough, 3, they're mature enough that they are basically ready for division. You have other proteins whose job it is to make sure that the division occurs when it is supposed to. And there's going to be checkpoints during the cell cycle where these proteins are going to do their job. Okay, there's feedback signals from the cell that can make the proteins start the next phase after it passes that checkpoint. So the first checkpoint occurs after G1. And this is where the proteins are going to make sure after the G1 phase that the cell is healthy and big enough before the cell is allowed to start the S phase, which is where the cell copies the DNA. That's the first checkpoint. If the conditions are not favorable, meaning the cell isn't big enough or it's not healthy enough, then the cell cycle will stop. It will not proceed to the S phase. It will not copy its DNA. This can happen, say, like at the last time that a cell divides, like I said yesterday, I think it's around 40 times that one cell can divide, because remember I talked about the copies of copies of copies of copies. If, let's say, after the 40th time and cytokinesis is just finished, and then the protein looks at the cell and is like, yo, bro, like you've already divided 40 times. So yeah, you're not clear to move on to another, the next stage of the cell cycle. So at that point, the cell can enter G0 because it's just not healthy enough. Sometimes the cell needs more time to grow. And so it might just be that the cell, what do you call that? Moving on, I can't think if there's a P word, like procedure, no, whatever. Going to the next stage will be delayed until the cell has time to grow bigger, or maybe it's lacking certain chemicals or something like that. The second checkpoint is after DNA is copied. And this is the G2 checkpoint. After DNA is copied, which you'll learn about in chapter 10, the steps to that's pretty straightforward. There's enzymes that check to make sure that the DNA was copied properly. If it passes, the proteins will signal the cell to start the molecular processes that allow the cell to enter mitosis. Sorry, it's not after the S phase, it's between G2 and mitosis. So I really love this graphic because this tells you exactly where the checkpoints are. So you've got checkpoint number 1, which is at the end of the G1 phase to make sure that the cell is healthy and big enough. If it's not, it can exit and go to G0. If it is healthy enough, it goes in a copies DNA and it prepares to divide. And just before it prepares to divide, that's where you get the G2 checkpoint. Make sure that the DNA was copied properly and it's ready to go. Once it passes that, you go through mitosis. At the end of mitosis, you have another checkpoint. If the cell passes, the proteins signal the cell to exit mitosis. You have cytokinesis and it goes back to G1. What happens if there's problems along any of these checkpoints? Well, what you'll learn in a couple of chapters is how genes code for the proteins and how that works. If a mutation happens in a gene that codes for one of these proteins may not function properly. Now, that doesn't mean, and what I'm going to tell you again in chapter 10, which is what I'll say now, cancer is a very complicated, convoluted illness. It is not simply like, oh, I have a mutation in one gene for one of these proteins that controls a cell cycle, so whoop, I've got cancer. No, because you have other genes that code for other proteins whose only job it is to make sure that these proteins are actually working properly. And you have other genes that code for other proteins whose only job it is to basically patrol around your body and make sure that cells are dividing properly. So even if you have a mutation in, let's say, the protein that regulates the G1 checkpoint, so maybe the G1 checkpoint doesn't work in a particular part of your body, you still have many other regulating factors that can prevent cancer from happening. Okay. It's also a question of your environment, your genetics. It's not just one thing or another. It's a very, like I said, a very complicated thing. So cell growth and division may be disrupted if there are mutations in these genes that code for these regulatory proteins. Disruption can lead to cancer, which is the uncontrolled growth of cells. Can, not will. Okay, can. It is possible. There are lots of people who have been as healthy as possible for their entire life. They've never smoked a cigarette in their life. They've never had a drop of alcohol or any drugs, nothing whatsoever. They eat a healthy, balanced diet. They exercise every single day, super healthy, and they still get cancer. There's also people that smoke for like 50 years and drink like a bottle of whiskey every night and God knows what, and don't care about their body at all. And they're perfectly healthy until like they're 100 years old. Like I said, health and illness is a very complicated thing. There are a lot of different factors to consider. Cancer cells do not respond normally to control mechanisms. So they can overproduce growth promoting molecules, which results in increased cell division. And you might also have interference with the control proteins that slow or stop the cell cycle. So there's a lot of, like I said, a lot of complicated issues. You might have cells that don't listen to the regulatory proteins and they're like, yeah, bro, I know you told me I can't divide, but I just really feel like dividing, so I'm going to keep dividing. Even worse than that is cancer cells can cause normal cells to mutate and become cancer cells. So that's even worse. It's like that phrase in English called one bad apple spoils the bunch, which is actually based on actual biological principles, which you'll learn about next year. But there is a gas that's produced by a rotting apple, and that gas causes other apples that are next to it to also produce that gas. So it really is literally one rotting apple will make the entire bunch of apples rot. And it's called ethylene. But anyway, cancer cells are the same way. right? A cancer cell is a bad cell and it can cause other cells to become bad. Okay, that is